if you want to get into the field.
I'm going to pull wide.
Okay, you can take bubble.
Thank you.
Dig in the Argus view. It's cool how that just ends.
Yeah, that is neat. It's like a little mound.
Yeah.
So it might just have a mound, Chris, and then it goes, and those the Palapagon sponges?
Poliopogon.
Poliopogon?
Yeah, there's a couple of them there.
Those were the dominant sponge we saw in the last dive when we were here. And sort of bamboo coral. Okay, let's go! Yeah!
Let's swing to the left and take a look at some of these big boys over off screen on the left.
Sounds good.
Are we doing our protocol?
Yes, we should do our protocol.
Okay.
There we go. We don't protocol, sorry.
Okay. So we are grateful that we have landed on this site to begin our 7th dive exploration. Before we continue, we are going to do our protocol. So this time we will be going radio silent to conduct a private ceremonial protocol to acknowledge and honor the biocultural resources that are present and that we are interacting with and impacting. So we'll be back on SPL shortly. Everybody on mute.
I think we're getting a little bit better at that. All right, thank you very much.
Sounds good, Mahalo.
So we do
Thank you, come wide, please.
Yep.
We do have some inhabitants that are still here, but not in the abundance that we were seeing above.
Definitely not.
Which is fine, because this is sort of a more normal abundance when you're not in this really rich place.
Was that a leaf sponge?
I think it is.
In the front?
I think it is.
Well done.
Good idea. And it's in the genus Tretopleura. There might be a little corallium, that little pink thing in the center.
Oh, yeah.
Yeah, so this wouldn't get us jumping up and down here like we were the other day. Going to take a look at this massive sponge.
Oh, what is this?
This is called Poliopogon again. There's a lot of them here.
What's the black stuff on it?
It's a shadowing, I think. There's your laser. It's about, 30 centimeters across, maybe? 40?
Oh. Oh, yeah, it was a shadow.
I'm going to tip the camera. So again, the lasers are 10 centimeters apart. So this one's about 30 centimeters across at the width. Or maybe more, 40. All right, can you zoom in, please?
Yep.
Let's take a look at those spicules, or whatever you call them.
So
wow.
This is a nice shot because it's got these what they call sieve plates. So it's this mesh over a lot of the holes in the sponge. You can see the holes, and it's got sort of a cage or a grid over a lot of these holes. And purpose can be to prevent animals from getting inside that could damage the sponge. That looks like a sea lily or something. I don't know what that was growing on the right there. And all of these sponges and a lot of the corals serve as homes for a lot of other small creatures. That's why it's so important that we want to preserve them, and so important they're in the monument as well, because you're not just preserving sponges, you're preserving sea lilies and arthropods and brittle stars and all types of things.
We see here on the backside here, it looks very dirty. Is this on the process of dying?
Not really sure what that is. I'm not. I'm hard pressed to think that's sediment, but it could be. No, I really don't know why it had that sort of brown appearance. But it's a healthy sponge. It's got a lot of white to it.
A viewer wants to know if this is one organism or a colony.
It's one
sponge.
one colony.
Great question.
but
That's a
it's
really
not really, it's sort of a multicellular organism. It's not like a bunch of actual individual animals that are then together. It's a lot of cells that are together.
So it's one organism made
One
of many
organism,
cells.
yes.
Sponges.
Yes.
Oh, and this is
And
a
then this
pretty.
is a totally different type of sponge.
Zoom in, please.
See the difference in these shapes. This is called Tretopleura, or the leaf sponge that Aphrodite, I guess, was guessing at. I think that was you who was guessing.
A leaf.
Yeah.
What is it?
Yeah, Tretopleura. So the one we just saw, it's what we consider a soft sponge. If you were to grab
I
it,
have a little more
soft.
zoom.
And this is a hard sponge. This would feel brittle.
And why is it called the Treatle?
Tretopleura?
Uh-huh.
I don't know. I don't know the origin of that name. What are the white things on the stalk? Are those barnacles, maybe?
It looks like it. Yeah, I can't get a lot closer, but
I can zoom in.
we can zoom
more.
more. Yeah, go ahead.
Okay.
See if I can keep it centered.
That's all I got.
Okay, I think they're barnacles, maybe, possibly, perhaps.
All right, come wide, please.
There's a definite answer.
Ooh, that's a nice.
Beautiful. Okay, so we've got some of these unbranched corals. Some of these may be bamboos. I think some of them are.
Some nice bigger corals there.
I forgot to log into the science portal. We'll do that right now. Maybe.
Oh.
So that's a big bamboo. We could, and there's some smaller ones too. Pilot, before we go, right on the left, going off screens now, is a coral that Scott France, right there, and he's right there. He thought this is here. He thought he saw one of these, and I think he's right. And I think this is one of the ones he thought he saw, if we could zoom it.
Okay, Aaron, zoom in, please. Oh, do not go down anymore.
That one.
Sponge right below me.
Yeah, I seem to have forgotten my password here.
Not the most easy to see in this view, but see what we can do.
Yeah, and
That's
if you
all my zoom I have.
can you look at the base a little bit?
Yep. You can stay in, Aaron.
Okay, cool.
That's got to be it.
It's bare.
Yeah, but it's the branching. You know, I have no idea what's going on here, but it has not taken my password. Anyway, this could be no, it's not Acanella weberi. It's something else. Okay, thank you very much.
Okay, come wide, please.
Yep.
Okay, well, would you like to keep exploring this little mound, or should we start moving? It'll take a minute for the ship to start moving.
Yeah, how about that bigger coral up there? Let's take a look at that one. It's telling me it's an invalid password, and I know it's right. So
Okay,
Oh.
zoom in, please.
Yep.
I don't know what's going on.
I don't know, but there's
keep
no
getting a
other
little
comments
closer.
inside chat right now.
Mess with your
Oh,
focus.
okay, maybe I'm going to look over your shoulder.
Yeah, I'm
Sorry.
still logged in under Steve's login, so
Okay,
I'll tell
this
you if
is-
anything comes up.
This is a bamboo, and I think it's a nodal brancher. I think the branches are coming out those little black bands.
Yep, definitely.
And
can
that
see that
helps
pretty
narrow
clearly.
down its ID. We're looking at a, they call a basket star that likes to hang out on these
Can
things.
you come down in Delta a little bit, please?
Is it eating it too?
I don't think it's eating
'Cause
it
those
necessarily.
branches in the back look like, that it was on, look like they were stripped.
There's a little anemone on there or something?
Yeah, these are anemones.
Zoanthids?
These are anemones, and they're called amphianthus, is the ones that kind of cling on to coral branches. I don't think these guys eat the corals, frankly. I think they
Just get the height advantage.
get the height advantage. Exactly.
Okay, come wide, please.
Cool.
Thank you.
So Chris, do you know if any deep whip black corals have been collected as yet? Jeremy Horowitz is asking.
He's probably referring to these whip corals that are in the genus Stichopathes, and it's a single unbranched black coral. And yes, we have collected a number of those.
So
Okay.
if we're doing no collections here, we'd like to move along.
Yes, please.
Okay, we'll put a ship move in.
How about just 090 right towards there?
Sounds good.
Bridge nav. Can I get a 20 meter step 090?
Oh, I know what I'm typing it wrong. This is a senior moment. Good grief. There we go.
Yay.
Sometimes describe my brain as sort of the equivalent of an organic Cretaceous seamount that's all manganese crusted, and it's had some flank failures.
So are we looking to get some nodules as a sample?
Yes, please, if we could, because I don't know how many nodule fields we're going to find up.
You want me to do that right now?
Oh, sure, let's do that.
How big's that step?
20 meters.
That should be fine.
Okay.
I'll go to the far end of the field and we'll get a nice low delta and should be okay. Right?
Okay.
I think that'll be all right.
Standing by.
Okay, could you come down delta, maybe 8?
So viewer, can we ask a question?
Excuse me?
Yes.
Can we ask a question right now?
Yeah, go ahead.
Okay. A viewer wants to know what's the difference between a sponge and a coral.
Oh, big difference. Sponge is a much simpler animal, and a coral, it's in the family Porifera, or excuse me, the phylum Porifera, and a coral is in the family Cnidaria, which usually is used to describe animals with radial symmetry. You go, radial symmetry, that thing is branching, that's not radial. But each of the polyps on it, the individual parts of the colony, the colonial part of the animal, it is radial. They're round and they have a series of tentacles around it. So it is radial, just like an anemone. You know, a sea star is radial. It has arms going out in all directions. Sponges aren't like that. They're not radial at all. Corals are more complex animals, way more complex animals. Sponges are very simple. Some of the most simple multicellular animals that we have in the world right now. So those are a variety of reasons. There are many other ways that they're different, but that's it. The one similarity they have.
All right, science question, sorry to interrupt. My scoop in this stuff? Is that the right?
Or push core.
Push core or?
Is anyone around when Beth collected some of these last time? I wasn't on her watch, and I didn't see how she did it.
Okay, well I can like poke it if that's helpful.
Well, they'll come off a bit, but go ahead and poke it, and we'll make sure they're loose. Yep, they're loose. So, Aaron, you think it was a push core because you would scoop them? I don't think she really needs to get a lot of the sediment below it.
It's just the nodules on top.
Yeah, I think so.
Yeah, when I saw they do the push core, it looked like it was sand. It looked like it was fine sediment as opposed to this kind of field.
We've got a scoop, so
let's scoop it and just use one of the small boxes on the side.
Yeah, Roger. And if we're only going to do the one scoop on this dive, I would like to ditch the scoop in that box.
I think so. I don't think there would be any reason to scoop anything else.
This is Beth.
Hello,
Yeah,
Beth.
oh, hi,
Yeah,
Beth.
hey, Beth.
Hi. A sediment core would be great if we think it's deep enough.
Instead of or as well as
Hey.
scoop?
As well as.
Sounds good.
Okay, so we'll just scoop the nodules themselves and put them in one of the side boxes, right, Beth?
Sure, so you can put them in the side box, that's fine.
Okay, and then we'll try a sediment core. We'll try it anyway and just see how deep it is because we really don't know. But this is probably about as deep a sediment as we're going to find on this dive.
I'm digging the Argus again.
We'll give
Thanks.
it a shot. Yep, thank you, Beth.
So cool.
Okay, can you zoom on Herc, please? Thank you. So Antonella, this is the thinking about me dumping this before doing the scoop. That's why I'm doing this weird back-wristed thing.
That sediment looks reasonably deep so far. You're getting more sediment than nodules.
It's a lot of sediment there, isn't it? It's just so flowy. I'm going to sit that there for a sec.
It's super fine.
Need a sifter.
Yeah. Should have brought a colander down.
That would be fun.
I think it would go right through the colander.
So,
Pick a box, a
if
small
Beth
box.
is still watching, any input? Do you want more nodules than that? Like, should I be trying for something better, or what do you think?
Yeah, if this is going in the side box, the more nodules, the better.
Okay,
Okay.
so
This showers. This is gonna make a mess.
maybe I'll try. I'm just
Yeah,
gonna,
maybe a little bit thinner scoop or something, maybe. I don't know.
the nodules are just going to stick in there. That's great. That's
That is
fine.
pretty great.
Try to dump some more sediment out.
Cooperative.
All right, I'm ready to watch that.
Oh,
Oh, there they go.
Nice.
I'm going to try again. I'm just going to start fresh.
Yeah, I think that would be good.
You can come up on Delta now.
Whoa.
That sediment is thicker than I thought it was going to be. I thought it was more of a sheen, but I guess maybe nodules don't form if there's just a dusting of sediment on it.
No, because the water, the
Yeah, the
pore
water's
water's coming up.
That's right.
And then that's I mean, I'm going out on my limb here, but I mean, that's why those nodules are all, you'll notice that they're not stacked on top of each other. They're all separated by a little channel of sediment. And I think that's, I don't know, Beth, that's how they form that way, right?
That's all we get.
That's pretty good. That's all right. I think.
Okay, come wide, please. And can you put bubble on the starboard side? Yeah, probably.
Hey, Nalu, did you take some pictures of those nodules?
Yeah, I did.
Yeah, thanks.
Yeah, no worries.
Beth, you want him to try the push core right where he sort of cleared a spot there between the nodules? Would that be a good idea?
How do I turn this back on?
It's open.
Oh, okay. Can you hear me?
Yes.
Yes, we can.
It actually would be better to core through the nodules so that we can get both.
Roger that.
Otherwise, the sediment underneath is already too disturbed.
Okay, I got you, of course. Should have thought through it. Thank you.
Can you come up on Delta, please?
That's the voice of Dr. Beth Orcutt, who is out here. We talked about her before in her project out here as well. She's in the lounge, I think, so it was great that she's able to guide us on this.
Okay, I'll take sample salvo, please. Okay, actually stand by.
Okay, he's saying these steep whips are, yeah, I agree. These aren't stichopathies. We haven't seen any stichopathies.
Okay, box out, please.
Probably will be described and placed in aphanosticopathies.
I'm gonna go for alpha.
But what, these are, the whips that we're seeing right now.
Hey, sorry, can I interrupt?
Yes, please,
I
sorry.
want to confirm that I can dump this scoop in this box.
Yes, that'll work.
Fine by me.
Okay, great, thank you. Okay, close the box, please.
Roger.
Kira Mizella, who's very interested in these nodules, just sent a message here. She asked, how many nodule fields have we seen on this expedition?
And stop there.
And
Do you want me to bump it out?
we've
No, that's fine.
seen a few of them, I think, Kira, and I think we saw 2 of them on the upper area of this particular ridge that we passed over.
Oh, coral cutters and a sediment coral. Whose idea was this?
Oh.
So sloppy.
Hey,
Did you
Data,
chop
what's
the
our
shovel
sample
and
number?
That was sample 088.
088, thank you.
Oh, that's gonna spring
And then,
sprawling.
was that in box A?
Box A.
Can I fit it in here?
That was the closest, the furthest one forward.
Yeah,
okay.
box alpha.
Got
That's
it,
correct.
thank you.
I can go in there. I can do that. Okay, dive salvo, please. I'm not holding my breath that this is going to work so well because that's very flowy sand. But
so what
stay
are the
optimistic.
cores? What do the cores do? What kind of information are we getting from this core?
You can.
Where did
ask Beth. I think there's plenty.
Where did I scoop? Did I slide?
Excuse me.
Oh yeah.
Can you see it? Is it maybe?
I think it's in the starboard side.
Yeah,
see on the starboard
maybe
cam?
there.
Yeah, I must have turned.
That was weird.
I didn't even notice. Wow. Oh,
Oh.
oh no! Don't do that.
Oh no.
Light bar. Amateur.
Oh.
I like that. The self-cut there.
Yeah, that was not appropriate. Zoom in, please. Should know better than that.
And the answer here is no, this is not as easy as getting the toy bear out of the box at the arcade.
That one's rigged.
It absolutely, it absolutely is
Trevor
not.
would know if it's rigged.
Okay, the moment of truth.
See how
The moment
deep
of
it
truth, I think, is
goes.
pulling it out.
Okay.
And you know if it's
the pre-moment of truth.
oh, it's solid. Wow.
Oh, not that deep.
No, that is not.
It's only like an inch and
It's
a half.
moving the whole
2
vehicle.
inches.
I might shake this out and then try another spot. I'm gonna try something.
Okay.
Come wide, please.
So Chris, there's a question from Kira Mizell in the side chat.
Yeah, she asked how many nodule fills we've seen on the expedition. I answered it a few minutes ago.
Oh, okay.
I was saying we've seen few. I don't know exactly because we only know what we know on our watch, and I haven't gone through the photos.
But a lot.
I'm pretty sure we saw
Quite a few.
two. Yeah, I've seen two already on this further up on this ridge.
Yeah.
So I think everywhere we see sort of a flat area, we're getting some nodules.
All right,
Yeah,
let's
we've
try
seen
it.
it on every seamount pretty much,
Yeah.
I believe.
Take two.
All right, let's see the test.
Is 100% vertical down.
Oh, it's solid.
Yeah, that's not going to do anything for us. I'm not going to be able to hold that little in order to make it over to the side. It all falls out. It's already fallen out. So.
Thanks for trying.
Yep, you bet. I'll stow this and we'll be on our way, I guess.
So Beth, I think we're going to encounter another nodule field a little
Try and
further
shake
up
it
because
first.
there's another flat, if you will, on our way up. And if we have that, we could try again. But I expect they're all going to be like this.
Can I reset while you're sitting here?
We'll try.
Yes.
Yeah, so Beth, if you're still on, what are you looking at from these samples, from these core samples?
All right. You
The
can give me
purpose
the other
of
one.
getting sediment is to have as a comparison for the nodules in the rocks to see how similar or different the microbial communities are.
Oh, cool. Okay, thanks.
Okay, and we do want to make some distance today, so maybe we'll make
right.
a little distance until we see something.
You trying to rush me?
Collect. Oh no, I'm just saying. I'm just trying to give you a heads up.
I'm just
on
bugging
what
you.
to do.
You have 3 seconds.
Okay, fine. 2, 3.
Nice! I knew you could do it.
He put his gun back in the holster.
Okay.
I close it.
All right,
Can you give me dive, please?
Roger.
I'm going to do my little PSA here for since I work for NOAA and I work for Papahanaumokuakea Marine National Monument, which is where we are active today. So we are within the monument, which is also a World Heritage Site.
Okay, let's go.
20.
And
Sure.
we are initiating,
Bridge
NOAA is
nav.
considering
Yeah, 20 meter move 090.
this whole area as a designation as
Up
a
on the porch,
national marine
please.
sanctuary. And if you'd like to provide comment for that, it's up on our website at papahanaumokuakea.gov.
090. Thank you.
And we're accepting public comment through the end of January. And we're also going to hold some public meetings, 4 public meetings in December, which will be on Zoom. So anybody can participate from anywhere in the world. And the reason why we're considering this area as designation for a national marine sanctuary is that that would provide additional protection on top of what we already have as a monument. And the other protections that are in place for this region. And it'll also provide some additional permanence for some of those protections because the Marine National Monument was designated, it's a presidential executive order, and a National Marine Sanctuary is a congressional act which has longer lasting protections. So if you want to participate in that process or provide comment for this consideration, please go to papahanaumokuakea.gov and look for the sanctuary designation process. We'd love to hear from you.
So while we're waiting on the ship, let's get a zoom on this sponge, so stand by for that. I'm just going to get a little closer.
That's one of the sponges that attached to a rock that was a little too small for the current, perhaps, the one on the right.
Okay, go ahead and zoom.
I think he's going for the woolly one. This is a Walteria.
Yeah. Those are one of my favorites.
And all of these Walterias that have round pores on their side, we go to CF Lucardi, and all the ones that don't have round that just looks like a frame is oftentimes you go to Walteria Flemingi. And the actual genus and that particular species were named after a very famous cell biologist named Walter Fleming by quite a few years ago. So this side would go.
All right, come wide.
It was Lucardi.
Sure, yeah. I only got. Let's do that as the last one because we're going to do changeover.
It's wild, as you hardly see those. They're just almost completely translucent and disappear into the background.
Trevor, we just started.
I know. We're just getting warmed up.
We keep getting the blue water.
This is a black coral I'm seeing.
We're getting too good at
this.
this.
Zoom.
Yep, stand by.
Getting too good at this blue water stuff. They've been sticking us with that the last few.
Well, I hate to tell you, but
okay, go ahead, zoom.
I'm kind of sticking us with that.
Oh, you're the one. Oh, yeah, Bathypathes.
Yeah, Bathypathes probably, or let's see. Yeah, probably Bathypathes.
Black coral.
Yeah, now this isn't what Jeremy was talking about earlier. He was talking about the single branched type of whips that he said could be Phanosticopathes. But Jeremy, I don't think we've seen any of those on this dive. All of those whips are primnoids and bamboo corals, all the ones that we looked at. I would love to see some sticopathes here, but I haven't seen them. So it looks like they're not alternating, Jeremy, but it's an odd view, but I think this is more Bathypathes.
Well, ladies and gentlemen, we're changing over shifts. So.
Germany clearly knows something about black corals. So it's kind of nice to have somebody in the chat now.
Yeah, it's great.
Can help out with that group. And he's a new viewer, by the way, to the Nautilus Dives. And I hope we can treat you to some black corals, Jeremy, when we come across them. Another Poliopogon sponge.
Poliopogon.
You notice that the substrate here is not very consolidated. It's sort of rubbly and bouldery, and that usually does not result in a big community. It's very hard for some of these sponges to actually get settled and be successful on this type of substrate. We've seen this time and time again where when you get cobbly areas, you get a sort of reduction in the number of animals, and then when you start getting, oh, there's a crinoid, a sea lily in the back. I wonder if we could take a final peek, or are we done? I think we're done. So there's going to be a pause here. We're doing a watch change out right now, and so we got a lot of people.
Zoom in on the meaty bits.
Any chance you could get that crinoid behind it? Or is that too far away? I mean, just to zoom.
I'm landed right now.
Okay, got you. All right, thank you. Well, folks, you're going to get another bunch of voices to listen to for the next 4 hours, and then there will be another team coming in after that. So stay tuned. This dive has just started, and we've got a ways to go, and I think it's going to be really interesting. And I'm going to be watching. So
Okay, great. Thank
Yeah,
you for watching and following us on nautiluslive.org.
yeah, thank you very much, and aloha. Check, check, check.
Check, check. Full wide, please there, Jeff. Thank you. Good evening, ladies and gentlemen. We're waiting on a few more of our watch to settle in, and then we'll be going silent to ground ourselves before we get started with you this evening. Good afternoon, everyone.
Afternoon. I don't know what you're talking about.
Yeah, what time zone are you in?
Good evening. Yeah, that's better. All right, I think we're almost all settled in. Just give it one more second here. Thanks, Fernie. All right. Thanks for joining us this evening. As we do at all of our watch transitions, we're going to take a moment here to ground ourselves. We'll be going radio silent. We'll be conducting a private ceremonial protocol that acknowledges and honors the biocultural resources that are present and that we're going to be interacting with and impacting this evening. We'll be back with you shortly, and at this time I invite everyone in the control van to mute your microphones.
All right.
Thanks, everybody.
Hey there, Beth.
Got a zoomed out view of HYPACK here to just get an
thanks.
overview of where we're at.
Okay.
Just dove here. We've got a
intermediary dive waypoint, just up a couple contours to a flatter bit. And then I know that, from what I understand, there was some discussion about the path after that, whether or not we're going to this mound or up a different way, however you'd like to go.
Sounds good. Let's put it in a ship move to get us east over to that waypoint.
Sure. All right, we're gonna go 080.
080, Raj.
Bridge nav. Good evening. Let's have a ship move 100 meters, 100 meters, bearing 080. .2. Thank you. All right. It's looking like not too gnarly of a slope.
So as we're all settling in here, we're all kind of looking at our screens, checking in on what's been happening. We get a little bit of a preview as we watch down in the lounge. We're getting to watch the same views that you're all watching at home. But here we are. We've returned to the Don Quixote Seamount. Our intent tonight was to descend on the previous watch down to a depth about 2,100 meters, looking for kind of an area that we anticipated might have fewer, if any, sponge and coral communities. We're trying to kind of define the outer edge of the zone, the lower edge of that zone of habitation for the coral and sponge. And what actually did the previous watch find, Beth?
I don't know. I stopped in the lounge right when they showed up at a nodule field. So I know we've already seen some sponges, including one that had a lot of attached fauna, including a sea star. But I haven't been following along entirely. So we were just in an area that was pretty flat, kind of looked like the view we have right now, except less of these larger fist to grapefruit size rock pieces. It was mostly just small nodules. So we tried to take a sediment push core there, but we were still pretty close to the area where the sediment drape was just starting. So it was pretty thin. We were able to get a scoop of some nodules, but we did not get the sediment in a way that we can use for microbiology yet. But maybe we'll cross over something later in this dive.
Find a deeper sediment area.
Yeah. I think the key is to get as far away from the rocks as possible, so that the chance that the sediment drape is thicker is higher. But we'll see what we get.
It seems like and viewers have commented on this, that sometimes sediment seems to kind of collect behind rocks in kind of a deeper pile up. Is that the case because of the currents? Would you?
I don't know that the sediment is deeper there, but it just may be that the conditions aren't favorable for nodule formation. So it could be related to the currents in terms of...
Is there such a thing as sediment drift in bigger, deeper areas?
I don't know. I don't know enough about fluid dynamics of sediment flow around rocks. We also don't know the prevailing current direction down here yet.
Yeah, speaking of that, I was just taking a look at the video and looking at the little specks. And it seems like in this particular location, it's coming somewhat from the north to the south. But we'll see how that moves and eddies as we go up the feature. Looks like Argus just started to move.
For some reason, Rennie, I have you quite quiet again.
Check, check, check.
It's still pretty quiet.
Check.
Oh,
got
yeah.
I've got
That's
it.
good.
I can fix that.
That's better.
Is this better?
Yeah,
Yes.
that
Yep,
rocks.
that's better. We were millimeters difference.
Oh,
Just moved it like a little bit closer.
Yeah, Steve, any surprises thus far in just the first little bit here on the seafloor of this dive?
I haven't seen much in the way of surprises. I think it's been pretty similar biological community that we've seen on the dive a bit shallower last dive, but sparse.
Yeah, I know the dive plan kind of anticipated
sparse, yeah.
a sparse, lots of nodules.
Nodules are very hard for a lot of larger organisms to attach to. If you notice, like this coral that just passed us here is attached to the larger boulder in the slope. So not a lot of corals will grow big on nodules. So you have to wait until we get into this harder, more cohesive substrate.
Sorry for the radio silence. I'm communicating with some of our scientists ashore. Can't talk and type at the same time. Steve, can you remind us the name of these sponges that we're coming across that we're also seeing on the previous dive?
Yeah, so it's going to be sponge in the Pheronemattidae family, and we were tentatively IDing them as Poliopogon, but as Chris mentioned, that this group is kind of a catch-all, so it's not clear if that's how they'll remain tentatively ID'd after examination of the material we've collected.
Yeah, good to do a partial there, Jeff, while we wait.
So stay tuned.
It's great there, thank you.
It's quite a hold fast.
Yeah, very cool.
So for our audience at home, if you don't have the Hercules data in front of you, we're at about a depth of 2,130 meters. So quite a bit deeper than our previous dive.
All right, full wide, please.
On the same flank, just the other day.
Coming up a little bit there.
Yeah, and we really are quite close, relatively, to that previous dive, the very last dive that we were on before we took a couple of days off to sonar map. We are still on the northeast side of Don Quixote Seamount.
Northwest.
Sorry, northwest. And I'm sorry, I was reading and speaking. In fact, close to the endpoint may kind of align with our previous dive a little bit and even overlap it.
Even just now, we're starting to accumulate some more coral species as we move into this larger boulder type environment. So we've added Candidella and Paragorgia to our list of octocoral observations. We saw some bathypathes with very heavy quotes on that. from a bit earlier. That's a black coral. And we'll see what we see here. We noted probably upwards of, in the thick of the high density, high diversity community we were a few days ago. We were up to probably somewhere in the 20 to 30 species range of corals. But of course, to do a proper census, we would have to do more collections and more tight zooms on things to get a better handle on the IDs.
Is that Walteria?
What is that laying on the rock there?
A dead sponge.
On the left, with the pink?
Yeah, I think it's one dead sponge and one maybe live sponge with some attached associates.
Yeah, oftentimes.
Oh, we've got a fish.
Cutthroat eel there, maybe with a parasite or some sort of scar towards the head end.
It's a fairly clean looking, is that Walteria? Is that the name of it?
On the right?
Oh right? Yeah, on the lower right, yeah, that's Walteria, glass sponge.
Seems to be less stuff on it.
Thank you, Jeff, for already having the telestrator queued up so that the audience at home can see my beautiful penmanship.
Go ahead and fish on in there a bit, please, Jeff.
Brittle star's on the move.
Mm-hmm.
Yeah.
It's like a partially crusted stalk there on the top left.
Yeah. Come a little wide there, please, Jeff. Go ahead and fish on a little bit there.
This brittle stars cannot bend and chip.
Monkey bars.
Out of there.
Just kidding. Oh, wide, please.
They tend to do that, and I can't give you a good reason why they do that, but I think they sense maybe a pressure wave from the vehicle, and it wasn't uncommon during the capstone exploration expedition years where we would approach coral colonies and they'd be covered with these brittle stars or sponges even, and they would just all fall off 1, 2, 3, 4, 5, 6, and I don't know why they do that. They probably work very hard to get up into those colonies.
Mm-hmm.
I wonder if it's either sensing a predator or more like, I wonder if they ride any kind of current to go somewhere else. And
yeah, I wish I knew.
That was 2017, is that right? The capstone?
Yeah, 2015 to 2017 was the range of capstone. That's the campaign to address Pacific monument science, technology, and ocean needs. It's a big long.
I never knew what that stood for. Thank you.
I think they came up with the word first and then backfilled.
Yeah.
Isn't that how you come up with any good acronym?
Yeah.
Would the noise of Herc have anything to do with that? With them deciding to, because Herc is not quiet underwater.
No, definitely not.
Yeah, maybe there's a vibration associated with that.
A little
Interesting
further
reminder.
out in front of you here, Jake.
What was that?
So I'm going to get a little further out in front of you here. Feature. Sorry, what you're going to say, Beth?
Oh, no, that was me. You're good.
Oh, sorry.
We have a question, maybe Maybe Renato, you can answer this. Someone's interested in making a 3D print of some of these seamounts. Is there any way to access the high fidelity maps of these seamounts?
Yes, actually. It will, all of the data we collect will be publicly available. If you'd like it earlier than it gets placed in public repositories, then you can, I believe that there's contact information on Nautilus Live and or the Ocean Exploration Trust website. But barring that, if you'd like to get some slightly older data or wait for this one to come out, we upload everything to R2R, Rolling Deck 2 repository, where it's archived. There's a few research vessels that contribute to that. And then from there, other entities grab that information and put it into other archives such as NCEI, which is the National Center for Environmental Information. So if you're interested in any kind of, at least on U.S. waters bathymetry, you can get raw data and whatnot there. And eventually it ends up in Google Earth as well.
Yeah. There's also the MGDS, and sometimes data is, I think, uploaded to Pangaea as well for European data sets.
And general bathymetry, that kind of is like a rolling up-to-date map of everything that has been globally covered, is JEBCO and why am I forgetting the other acronym? I use it all the time.
The Seabed 2030?
Yes, that is a project, but why? It's like, let me embarrassingly give myself a second.
That's cool, yeah, and you can send that request if you wanted these particular seamounts that have just, in the last few days been more detailed, are mapped in higher definition. These, you could send a request to info@oat.org. That will eventually get to the right people.
GMRT. I don't know. I say that 10 times a day. I don't know why.
Off to the right-hand side, can we? Is
Right
it okay?
here?
Do we have time to take a look at that bamboo coral?
Yes,
sure thing.
I think we do have time. That's the whole point of this, is to see where the species start.
Yep. This particular colony was a request from one of our participating scientists ashore, so they wanted to get some really good tight close-ups on the coral colony and where it branches.
Roger that.
On the branch specifically.
Yep.
Great.
I'll park then for you guys.
Gonna do a DVL reset here.
Roger.
Yeah, just to let you know, the ship move is just about complete.
Okay,
Argus has probably another 20 meters to swing.
Okay.
But we could potentially be here for a little, although there is some topo.
Steve, could you elaborate on the interest in this branched species and kind of what the what our scientist ashore is interested in finding out?
Yep.
Ready?
Yeah.
Yeah, so this is a bamboo coral, and the bamboo corals are a bit of a
Go ahead,
conundrum, especially certain morphologies. Yeah, there we go. I don't know if this is the exact one, but it resembles one of the sparsely branched colonies that that one of our scientists ashore, Scott France at UL Lafayette, is interested in. And basically, they're trying to find out, can you distinguish different species based on branching pattern, and when might corals that grow in branching have a branching morphology? When do they branch in their lifespan?
Okay, go ahead, Ernie, again.
Because it can be sometimes difficult to determine if, from video, if a coral is an unbranched or a branched colony. And so by getting good information of these branched and unbranched colonies across their various different growths, helps us better constrain those identifications from video.
Sorry, guys.
We did collect one of these the other day, so we have a representation from this site, but the video and just the observation is just as important in this case as the actual collection.
All right, go ahead, Jeff. Thanks.
And while this is a sparse brancher, what I'm seeing now is that this particular bamboo coral might not be exactly the one they're looking for, but I'd like to get some images.
Yeah,
anyway.
no, I can't help to have a comparison.
Yeah.
A little wide, please.
Is it the places where the polyps come out are staggered on this one? Is that, but we have seen some where they're right across from each other too, right?
Yeah.
I'm sure there's a technical word for that, but I don't know what it is.
It's more, what they're looking at is actually more of where the colony branches. So does it branch on a node or internode? First step. Second step is the colony morphology when the polyp morphology when the polyps are retracted and contracted. So this one seems to be branching at the internode, but it's branching at the wrong place on the internode. So the colonies they're particularly interested, first of all, branch just above the node.
Sorry, guys, I have to go.
Or just around the node, not exactly on the node. That's also another diagnostic characteristic. That's great, thank you.
Sorry about that.
Yeah, no, that's perfect. It wasn't exactly the colony we're looking for. And, you know, these types of things are a bit nuanced, but some of the things we're looking for discriminating bamboo coral species are both, you know, branching structure, but also colony morphology and polyp morphology. So these are very subtle differences that help us tell if, you know, if that's the coral we're looking for or not. But the polyps there.
Another fish here with that same marking on this.
Yeah, it's a little bit more pronounced than the last one we saw.
Yeah, interesting. If you want to go ahead and push on in there for a brief moment. Okay, full wide, please.
Looks like Argus is kind of seeing the flatness here that we're anticipating in the bathy.
We're starting to get some larger primnoid colonies too. These ones on the left and the right we saw a bit ago, these look to be primnoids. Probably in the genus Norella, what we determined last dive. They tend to grow quite large. But you can definitely tell things are starting to pick up in density a little bit. You're starting to get these thin whips, like the one immediately behind this larger fan, which is most likely Candidella, another primnoid that we noted from our previous dives.
Oh, look at all the brittle stars on that one.
But at the same time, there's also some unbranched coral colonies that are bamboo corals that were actually among the densest corals in our previous dive.
There's a large stalk there.
Yeah, look at all the stuff on that stalk.
We haven't spent a lot of time on the steeper terrain here, but it seems like we're seeing a lot of population density on the flatter, the less steep slopes.
Yeah, I mean, it's where you find high density life on seamounts is a combination of many variables, from water flow conditions to substrate characteristics. It's almost like you have to have the perfect conditions to have the high density communities we observed previously. And those conditions seem to be right at this particular slope. Sometimes steeper slopes are more favorable, and sometimes these more gentler slopes are. So we're still learning a lot about how the seamount landscape can either control or dictate what types of diversity and density of corals and sponges and other attached organisms you might find.
It seems that they are also preferring these more sheet-like flows than the pillow basalts.
Yeah.
I'm going to add another move, but I think we've got some time to look around here for some zooms.
Zoom, zoom. Steve, do you have a preference on what we spend some time imaging?
No,